GATB 2020 is knocking at your door and the deadline is approaching. You suddenly wake up and found yourself inside this sea and you don't want to drown. You want to fight back and ace this exam. So this particular playlist is for you. This is called GATB 2020 5 days to deadline. This playlist might be a savior boat for you, which can help you to ace this exam. So keep on watching my channel and don't forget to subscribe and hit that bell icon for notification. Hi, in this lesson, we're going to solve last 5 years questions from JNU CEEB. Now this exam is known as GATB 2020 from this year, but we have to see the trend and analyze the trend based on 5 years question. And today's topic would be immunology. So let's begin. So I have collected questions from 2015 till 2019 and arranged them in a conceptual framework which would help you to understand the concepts as you go through this video. This video might be slightly long and it would be about half an hour, but at the end of half an hour you would learn a lot and revise the whole concept. So stay tuned and watch this video till the end. Before that, I wanted to say that I have also given combined entrance exam for biotech in 2016, and as you can see, I was topper in that particular year. My rank was All India 002, right? So let's begin and try to discuss and let's try to share the tips and techniques that I have used to ace this exam. Right, guys? So let's begin. So first question is, which of the following is not a lymphocyte? The options are A, T cell, B, B cell, C, natural killer cell, and D is dendritic cell. Pause for a moment and try to solve this question by yourself. Hopefully by now you have solved this question and the correct answer would be the dendritic cell. So let me tell you why it is the correct answer. So the question says, which of the following is not an lymphocyte? So what do we call as lymphocyte? So we know inside our bone marrow there is hematopoietic pluripotent stem cell, and hematopoietic pluripotent stem cell develops into two different lineage, the lymphoid progenitor and the myeloid progenitor. So all the lymphoid progenitor derivatives leads to lymphocyte, whereas the myeloid progenitor give rise to many WBCs, RBCs, etc. So definitely the dendritic cell is not a component of the lymphoid progenitor cell or a descendant of lymphoid progenitor cell. So that is why it's not a lymphocyte. Now dendritic cell is unusual because it can be generated from both myeloid progenitor and lymphoid progenitor, and that is why it is unusual. Next question says, which of the following is not a hallmark of secondary immune response? It is rapid, it is more intense, it is due to activation of naive lymphocyte, it is due to amplified population of memory cells. Pause for a moment and try to solve this question. By now, I think you have solved this question. So let's try to understand the concept. The answer would be C. Why? In a moment, it would be clear that secondary immune response is way more rapid, intense, and involves memory cells, and it is way stronger than a primary immune response. So let's begin to understand a primary versus a secondary immune response. Notice that the primary immune response has a lag period, and that lag is pretty long. So here is the pathogen exposure, and after the pathogen exposure it takes 10 to 12 days for generation of IgM antibodies. After that IgG antibodies are generated. But in case of a secondary immune response, when there is a second time of pathogen attack, then what happens? Look at the lag. The lag is low and the response is higher. Look at the IgG curve. Its magnitude is higher. and also it involves memory cells. Now, if you want to get a better understanding of primary versus secondary immune response, click on the link in the I button, which is displayed on the top hand corner of your screen. Next question is about adjuvants. Adjuvants do not cause faster release of the antigen from the injection site, enhance co-stimulatory signals, prolong antigen persistence, increase local inflammation. So what are adjuvants? Adjuvants are simply those substances which can increase the immune capability or the immune response, which augments the immune response. So that is why it cannot cause faster release of antigen from the injection site. So that is contradictory to the definition of adjuvant, and that is why this option is correct. All of the following is a professional antigen presenting cell, except the options are dendritic cell, macrophage, mast cell, and B cell. So you have to find out which one is not a professional antigen presenting cell. I hope you have figured it out so far. So the answer here is the mast cell. So just a quick concept. Dendritic cell, macrophage, and B cell, all of these are professional antigen presenting cells because all of them present their antigen on top of class 2 MHC molecule, and that is why they are professional antigen presenter. Whereas mast cell do not express class 2 MHC, and it is not a professional antigen presenting cell. Next question says, which of the following is a primary lymphoid organ? Options are lymph node, thymus, spleen, Peyer's patches. Now let me tell you the concept of primary and secondary lymphoid organ. Primary lymphoid organ are those organs which supports immune development or the immune cell development, whereas secondary lymphoid organs helps in terms of supporting the immune cells. They don't directly help in the development process, but they help and support them and help them to mature. So the primary lymphoid organ here is thymus, where T cells get developed, and thymus is actually the training school for the T cell. If you want a detailed video about primary and secondary lymphoid organ, click on the link in the I button. So just to give you a quick overview, primary lymphoid organ would be bone marrow other than thymus, and secondary lymphoid organ could be lymph node, spleen, and many other places. Okay, next question says, which of the following statement is true? An antigen is always immunogenic. An antigen is always antigenic. An antigen can never be immunogenic. An immunogen can never be antigenic. So you have to identify which of the following statement is true. Slightly, this question is awkward. The correct answer would be B. So the option A is not correct. An antigen is not always immunogenic. Many antigens might not have immunogenic potential. We'll explain it in a moment. An antigen can never be immunogenic. So this is also wrong, because many antigens can also behave as an immunogen. Now, immunogens are generally antigen, so this option D is also false. So let me tell you, this is an antigen. So antigens are substances that specifically bound to antibodies or T cell receptor. So it can interact with the T cell receptor or the B cell receptor, that means the antibodies. Whereas immunogen can also bind to T cell and B cell receptor, but the difference lies in this particular thing, that they can evoke strong immune responses. Antigen may or may not evoke an immune response, but immunogen always does. So what we can say about antigen versus immunogen is antigen may or may not elicit an immune response, but immunogen would elicit a strong immune response for sure. Now antigens may or may not be complex, but immunogens are generally very complex in terms of their structure. So these are the quick differences between antigen and immunogen. Let's come to the next question. The basis of 5 classes of Ig molecule is number of amino acid residues, molecular weight of the heavy chain, structural differences in the carboxy terminal portion of the heavy chain, structural difference in the amino terminal portion of the heavy chain. Right? So let's try to answer the question. And if you have answered the question, the correct answer is this, the number of amino acid residue. So definitely all of these different isotypes are encoded by different gene segments. For example, IgM is encoded by mu, whereas IgD is encoded by delta gene segment, right? So their number of amino acids could be very different. Their constant domain, their hinge region, their valency, all of these things are different. And this is a fundamental understanding about antibody isotypes. Okay, contact dermatitis is an example of cytotoxic hypersensitivity, anaphylaxis hypersensitivity, cell-mediated hypersensitivity, immune complex mediated hypersensitivity. Slightly the names are inappropriate, but the correct answer would be here, option C, because type 4 hypersensitivity, which is also known as delayed hypersensitivity, is one type of hypersensitivity which involves cells other than antigen antibody interaction, and they requires quite a lot of time to develop, and that's why the name suggests delayed hypersensitivity, and it's the only type of hypersensitivity which involves T cells and no antigen antibody interaction, but the name suggests it takes at least 48 hours to develop. Maturation of the B cell takes place in the following order. So we have to identify the correct order. So let's try to understand this question carefully. The question says progenitor cell, affinity maturation, Ig gene rearrangement, class switching. So these are events in the lifespan of a B cell. So you have to understand which event came first, and you have to order these life events of a B cell. So the correct answer here would be option B, and let me tell you why. So first, B cell development starts at the bone marrow, which is a primary lymphoid organ. Inside the bone marrow, there are lymphoid progenitor which give rise to late pro-B cells or the pro-B cells. These pro-B cells eventually become pre-B cells and immature B cells. Now in this pre and pro-B cell stage, the gene rearrangement also takes place. In the immature state, they are secreted into the bloodstream, from where they can move into the secondary lymphoid organs such as lymph node. Point to be noted that in the pre-pro stage or in the pro stage, VDJ recombination takes place in the antibody gene segments, which leads to antibody diversity. So VDJ recombination is the next step in their life event. Now after that, they mobilize to secondary lymphoid organs such as lymph node, and inside the lymph node, in the specific region known as germinal center, many things happen. So after they interact with the T cells inside the lymph node, somatic hypermutation takes place. Somatic hypermutation is another way of generating antibody diversity. If you want to know more about somatic hypermutation, the link is present in the I button. Click on the I button right now. Now, somatic hypermutation ensures all the high affinity receptor containing B cells survive and others should die. For example, this B cell had a very low affinity B cell receptor and it would die. But if the B cell have a very high affinity receptor, it would survive and it would differentiate into plasma cell. Initially, plasma cells secrete IgM or IgG type and IgD type antibody. But eventually class switching occurs from IgM to IgG class or many other class such as IgA and other IgE types. So class switching is the last event in the maturation process of the B cell. So I hope this concept is clear. Which class of B cell receptors are expressed in the naive B cell? Naive B cells are immature. They haven't encountered pathogen, and that is why class switching has not occurred in them. So the prevalent receptors that is present on the naive B cell is membrane-bound IgM, and very little extent IgD could be also present. So the correct option for this question would be IgM and IgD. So let's talk about the next question. It says super antigen can bind to MHC class 1, MHC class 2, TCR and MHC2 together, BCR and MHC2 together. So the correct answer is option C. Why is so? Because these super antigens, which are also known as bacterial endotoxin, or they are actually a subcategory of bacterial endotoxins, which can laterally bind to TCR and MHC class 2 and lead to a non-specific activation of the T cells, which is like a false alarm in our body. Okay, let's talk about the next question. CD8 is a marker for B cell, cytotoxic T cell, helper T cell, and an activated macrophage. It's very easy. The question says what CD8 marker stands for, and it's a co-receptor for cytotoxic T cell. So just to give you a quick idea, inside the thymus, development of the T cell takes place. Inside the thymus, the double positive T cells, which has both CD8 and CD4 co-receptor, might either recognize the antigens present on class 2 MHC, or it might recognize the antigens that are present in class 1 MHC. If it recognizes class 2 MHC, it would be a CD4 helper T cell. If it recognizes class 1 MHC bound antigens, it would be a CD8 positive cytotoxic T cell. If it recognizes nothing, then it would be dying due to neglect. But if it recognizes both of that, then there is a problem, and there would be negative selection, which would kill both of these cells which recognize both which recognize class 1 MHC and class 2 MHC. Okay, so let's talk about which one is the characteristic of B cell but not a T cell. Class 1 MHC, CD8, CD3 receptor, FC receptor, membrane bound immunoglobulin. So obviously membrane bound immunoglobulin is a distinct character of B cell, right? And membrane bound IgM is found in naive B cells, right? So this is a characteristic feature. Which of the following cell would kill when MHC1 expression is very less in the target cell? And the correct option would be NK cell. Because the way NK cell work is very fascinating. NK cell interacts with the class 1 MHC bound peptides. So each of the cell type of our body express class 1 MHC. So it tells the NK cell that everything is all right if they display self antigens on class 1 MHC molecule. But many viruses suppress these class 1 MHC expression. Now, if the NK cell see the target cell has quite a lot of MHC class 1 bound peptide, then an internal signal would be relayed which says disengage and don't kill. But if the other way around is true, that means if there is no MHC class 1, and that is why there would be a different signal which says engage and you have to kill that. So that is how they work like a cold blood killer and kill those cells which down-regulated MHC class 1. Okay, the class 1 MHC pathway primarily leads to, so it would tell you what this pathway is important for. So this class 1 MHC molecule, based on this particular option categories, the correct option would be this one, because class 1 MHC molecules sometimes display viral antigen which could be displayed on class 1 MHC, and it would be displayed on the surface of the target cells or viral infected cells. So let's say a cell is infected by the virus. So the proteasome inside the cell would degrade many of these viral protein that would be packaged inside the ER on class 1 MHC, and it would be displayed on the surface, which could be detected by CD8 T cytotoxic cells, right? So the major event here is loading of the viral antigen along with the self antigen on the class 1 MHC, and several transporters and chaperone molecules help in this process. And ultimately, it helps to present these viral antigens onto to the cytotoxic T cells, right? And which would lead to the apoptosis of the infected cell. So interaction between dash and dash are not restricted by MHC molecules. T helper cell and the dendritic cell, plasma cell and the target cell, then CD8 positive T cell and target cell, TH cell and B cell. Now, based on that, what they are saying that all of these cell type requires MHC molecule interaction. For example, dendritic cell is a professional antigen presenting cell, so it would have MHC class 2, right? And T cell would have TCR, so this interaction is there. Whereas the CD8 positive T cell also has TCR and CD8, which would interact with MHC class 1 bound viral peptides. TH cells and the B cells. Now B cells always have MHC class 2, which would interact with TCR of the T cells. So the leftover option is plasma cells, because plasma cells secrete antibodies and these antibodies are soluble, so there is no MHC interaction. It would directly move and bind with the target antigens on the target cell or a pathogen. So that is why option B is the correct option in this case. Okay, then they ask interferon beta is produced by what type of cells? Bacteria infected cells, virus infected cell, both bacteria and virus infected cell, or fungi infected cells. So generally these kind of interferon are generated upon viral infection. If you want to understand how body fight back against virus, so you have to click on the link in the I button. Now let me tell you in short that the infected cell secretes interferon, which is an alarming signal to the neighboring cell. The neighboring cell knows that the adjacent cell was infected by a virus. So the neighboring cell has some amount of time to prepare itself and create antiviral molecules which would help them to combat the pathogen, right? So this is the important part. Now, what happens is the infected cell secretes interferon, which binds to interferon receptors and lead to JAK-STAT signaling. This JAK-STAT signaling ultimately leads to production of several antiviral molecules such as PKR, which binds to the viral DNA and inhibits translation. Also, there is 2,5-oleic A synthase, which binds to viral dsRNA and lead to its cleavage with the help of RNase L. Then there would be MX proteins. MX proteins can interfere with viral assembly. So interferon is an interesting and important concept. If you want to learn more about interferon, click on this particular link in the I button. Viral replication within the cell is inhibited by tumor necrosis factor alpha, interferon alpha, IL-1 and IL-4. So the correct option is again interferon alpha, because interferon interfere with viral assembly, right? Then the last question is bone marrow transplantation in immune compromised patient can potentially cause, so these cells are, this individual was immune compromised, right? Here the options are GVHD. GVHD means graft versus host disease, T-cell leukemia, delayed hypersensitivity, inability to use live donor. Now let me tell you, the option A is very alluring, but this is not the answer, because the graft rejection happens when our own immune system treat the graft as a foreign substance, but here the individual is immune compromised, so this option cannot be true. Now you figure out after listening to this whole lecture which option would be true and let me know in the comment section. So this was a comprehensive summary of last 5 years question from immunology section in GATB 2020, JNU CEEB, otherwise known as GATB 2020. So if you like this video, give it a quick thumbs up and don't forget to like, share and subscribe. Do let me know in the comment whether you want more videos of similar kind, whether this video was helpful or not. So stay tuned and keep on watching. Thank you.